I'm going to tell you, you've heard every single one of these things before and these concepts because you had to have these prerequisites basically to get here. And like I always tell people, I know you've seen this and so you get, you know, but if you are hearing something more than one time, or like this kind of stuff where you've heard it 8 times, there's a reason you're supposed to know it. And so every time you hear it and you see it, you get it a little bit more, or because you do have that sort of basic understanding of it. So hopefully this will just help you kind of have a few concepts click a little bit better. Oh, speaking of click, I get to use this. Woo-hoo. Do you guys have any questions yet? Good, I'm doing great. All right, I forgot which button is that one. Yeah, it is. Okay, the human genome. Let's begin there as an overview. Because it's genetics and we have to talk about the genome, which is the DNA. And this is why I freak out about DNA. I love it, total nerd chills every time, every single time. And the professors that taught me this stuff when I was an undergraduate and graduate school were not taught this stuff because it's a new discovery, really. We knew about some sort of, certainly Gregor Mendel knew about some stuff, some elements that were transposable, and that was very well understood, but the structure of DNA was only discovered in the mid-50s, or mid-century, from Watson and Crick, and so that's really recent history, and so then you fast forward to all this stuff about DNA, and there's every single show is, I mean, okay, maybe you don't watch every single forensic show, but every show I watch is, you know, they're looking for DNA and they're trying to get in, you know, so we just accept DNA. But it's so beautifully, universally simplistic and so elegant, and absolutely every biological thing on the planet has DNA as its source, if not just a nucleic acid, because there are some RNA viruses, but still nucleic acid with its 4-letter language in every living thing. Things that are extinct to now. We share this with dinosaurs, you know, we share this with Jesus, we share this with all the animals that have ever been. And so I think that's just so, it's so mind-boggling, it's so beautiful, it's so elegant, and it's the thing that makes me think, oh God, do you feel me? Do you feel me on it? It's exciting. And so, then it gets more complicated. But the only reason that DNA makes anything biological different from another biological thing, why we're different than frogs or funguses, some more different than others, is the fact that DNA is the recipe book for the proteins that are manufactured by the cells in that body. And those proteins alone are what make every cell, and then every multicellular being look different, act different, have its different job, have its different life. And so we're so only concerned with ours. You know, I come from a zoological perspective, so I just get excited about this stuff. I can't wait to start my shift. God, I hope you know that one. Okay, so all the genetic material or DNA in the human cell, and so what happens in the cell is what happens in the body. So we are really just orchestrating all the little individual cellular things. And so all of our cells have the same DNA in them, and that's the human genome. We have 23 pairs of chromosomes, you know that. And how much difference is there between us? It's about 1.0%. I picked that little line up of kids, sort of prepubescent, and you can see that, you know, things are different all along the way, as we know. And our nearest relative are chimps, and we share all but 4% of our DNA with them. And that's another miraculous thing when you go to Oklahoma City Zoo, for you that aren't from here. We really have a nice zoo, a really good breeding program and lots of primates. And you can just, I held my baby up to, it was a gorilla, and she held her baby up to me, and so there's just this, she felt me, she got me. So anyway, I get excited about DNA. So, oh, your favorite words, diploid and haploid. Do you remember the first time you hated those words? And you still do, right? I mean, you do, right? You just don't want to talk about it. But diploid is 2N, D, die, by, dip, 2. 2N, where N is one complete set of chromosomes. So one complete set of chromosomes for humans is 23. all of our cells have 2n, which means 2 times 23, which isn't the same as 46. That's why you hate it, right? One entire set came from the maternal side, and one entire set came from the paternal side. Those sets together make each cell diploid. And so we have 23 pairs. So for chromosome number 1 from your daddy and chromosome number 1 from your mama, they're homologous pairs or similar chromosomes, and so they're a pair. And so we have 23 pairs of chromosomes. There are some cells that only have one set of chromosomes, and those are the gametes. Your mama's cell and your daddy's cell, the sperm and the egg. Ooh, we don't like to think of them that way, though, do we? I mean, we don't mind ours, but our parents not so much. So we think they just passed by each other. There was a sip of wine, and the next thing they knew, they woke up, and you didn't see it. None the wiser. But for us, it's totally different. I'm sorry, was that inappropriate? Because I kind of hope so. It's genetics. The only reason we study genetics is because we are sexually reproducing creatures, because there are two flavors of gametes, egg flavor and sperm flavor, right? So gametes are created through a special kind of cell division named meiosis, and you didn't like that either, I'll bet, because it was that one with meiosis 1 and meiosis 2, and we'll get there. And so the somatic cells, soma means body, and so those are the body cells. So all the other cells except gametes and the gonads are going to be diploid. So that's the natural state. Those are the cells that are making the proteins that are making the body. They're called somatic cells. And those cells undergo the cell division, mitosis, which you notice. Look at this attractive couple getting ready to fertilize an egg. Hey, well, we're already naked. I think that's funny stuff. Okay, so right here, her big old egg and his little old sperm make up, those ends combine to make up a 2-end, haploid to diploid. That's all it is. And then all of those cells undergo mitosis a bunch, and then you get these whole bodies, and then they mature, develop into puberty, and then they make their gametes. And then they like, hey, we're already naked. Yeah. So the circle of life continues. So, please stop me for questions. So we're going to look at the genome itself a little bit. DNA is huge, lots of information there. But not all of the DNA is going to serve as a recipe for proteins. So those are the genes that serve as a recipe for proteins, and a human cell will have in the neighborhood of 20 to 25,000 actual genes. And that's only 2% of the genome. So there's a lot of DNA there that used to be called, very recently, I was taught it was junk DNA. And then some people did some research and were like, okay, maybe it's there for a reason. And it is, so we'll see what that is. But here's what DNA is doing. We'll look more at this, but you have the double-stranded helix in which there are genes. They are transcribed into a segment of messenger RNA. You're remembering these things. And then that is translated into an amino acid sequence. And so a gene is in the neighborhood of an average 1,000 nucleotides in length. And a protein may be in 300 amino acids. But that's just, there's a wide range of sizes and so forth. Oh, and we are cells that have a nucleus. So you see that this is actually an excellent diagram to show you that the DNA is in the nucleus. And we don't send the DNA out to the cytoplasm for protein manufacturing. We just make a copy of the page we need and send that out to the kitchen to make the proteins out there in the cytoplasm. Okay, so genes, and then non-coding regions. So what's in there? You've heard the word intron before. That there are segments within an actual gene that don't actually encode for protein. So they're the parts of the recipe that get cut out, the introns. And those are non-coding regions within a gene. And they're moved during RNA processing. That's Oklahoma for processing. And I believe I might have added that since I looked at these slides. So I didn't change much here. So there's your little DNA. I just keep adding pictures because I just love it. It's pretty. There's a chromosome. When you unwind it, you see a segment of a DNA with it. It's a gene with its introns and exons. The exons are kept. So all of it is transcribed. And then mRNA, messenger RNA, is processed pretty heavily there in the nucleus, and sections are removed. Those are introns. There's intergenic DNA. So intra is within, inter is between. Intergenic DNA then is bits of nucleotide sequence which are just between genes. And so probably a lot of them serve as sites where there is information for control of transcription or maybe even replication. So it's information for how to use the gene probably, but it's not coding. So coding means that you end up with a protein. There's also a lot of functional RNA. So you make copies of DNA in the form of RNA, and functional RNA then gets used for other things. But it's not a gene, it's RNA, so it's considered non-coding. So the next slide is, what are the different flavors of functional RNA? Why, here they are. More of the non-coding regions. Ribosomal RNA is one. You've heard, you know that one, rRNA. That's used during translation, the actual protein manufacture. Transfer RNA, also used during translation for the actual protein manufacture. And I'm saying these things, and I'll repeat these definitions again later for transcription and translation if you're not holding on to what those mean. We'll go over it again, a lot of redundancy here. There is also some of those little RNA segments that you kind of heard about. Now, if you studied a good bit of cell biology during your undergraduate, then you heard a lot about it. If you didn't, then that was kind of a in your mind, it's under a parentheses, like and other RNAs. But there are these little bits of RNA. They're in the neighborhood of 20 to 35-ish nucleotides long, so quite short, and they have other functions. So they get transcribed like any RNA does, but then they hook up with proteins like most RNA does to become functional. And then those complexes, the Pee-wee interacting RNAs is an example, piRNA, they hook up with Pee-wee proteins. That's a segment of proteins in the cell called Argonaute proteins. So there were some nerds naming that stuff, right? Like me. I'm not putting them down. No judgment here. But anyway, Argonaute DNA and the proteins, and they will combine with RNA. And what I'm trying to get to say is they're regulatory proteins. And so they're going to regulate, especially post-transcriptionally, or that is to say, when messenger RNAs have been made, they can help destroy it. The RNA can recognize and complementary bind to RNA, and then the protein complex is there, and really just inhibit translation, inhibit the gene product, in other words, silence the gene. So Pee-wee interacting RNA and microRNA, both short nucleotide, RNA nucleotide segments which combine with proteins to create gene silencing, mainly acting at the level of messenger RNA. And so when you think about it, think about this. This is another one of those areas I really like. You didn't like embryology though. You studied hemophilia. Or is that just me? I'm just thinking about the things that I first learned and didn't like. I feel like they're kind of universal. But embryology was a bit tough. When you think about something going from one fertilized egg to, this, it's a big, that's a lot of stuff has to happen. So there are a lot of genes that are on embryologically to create mitosis, and then the movement of where these cell layers begin to form, and then arm buds, and then an eyeball, and all of that stuff that happens, all that great differentiation. There are a lot of genes that regulate that happening early on that need to be turned off. And so a lot of those genes are turned off, but what if they come back on? It's not good. That's actually, we see that as cancer. Cells forget who they are, they regress developmentally, and then they just think there's some cell in an embryo wildly undergoing mitosis. So there has to be a lot of downregulation of cells, of genes that shouldn't be on. And that happens at a lot of levels. One of the levels is post-transcriptionally. So between the gene and the gene product And RNA itself helps clamp that down and turn those off. So there are only 20 grand for proteins, so few of those actually make up this body, and a lot of them are regulatory proteins early on. So it's just a miraculous feat, and there's lots of bits that accomplish it. And these Peewee interacting RNA bits and microRNA bits do that as well. So it's part of what the purpose of DNA is to control itself. Don't we have to have self-control? I mean, we can parlay all of this straight up to us, you know. But it's sort of this self-control. So you have to have it all there so it can go, but you have to really be able to inhibit it a lot. And what you learn about all the way through physiology and all of this stuff you're learning is when disinhibition occurs, disease states arise. And so that's what you're really studying, is what doesn't, what doesn't go right. Whew. Exciting. You have a question?
I do have a question. Is microRNA and messenger RNA, is there like a nomenclature, shorthand way to differentiate the two?
Oh, yeah. You're right. That's my error. It's miRNA.
Oh.
Thank you. Change that. Yeah. It's miRNA, like piRNA. Good call. Because that would really mess up the world. Although there's some, that's some of what they do at those meetings when they get those scientists get together and drink beer. They're like, how can we screw these kids up? Pseudogenes are genes that look like they would encode proteins, but they don't, and they are thought to have kind of an evolutionary past. So they're vestiges of DNA that isn't gone yet from the species, but the genes are no longer, the proteins are no longer needed in that species. So there's some sort of, that's kind of interesting evolutionary or relatedness sort of study. Cis and trans segments. Do you ever watch Caitlin? Caitlin, what is it? Bruce Jenner? Caitlin, there's a show. I Am Kate. I have a great fascination with that. And anyway, they're transgender, which means different gender, and their cis means similar. And so heterosexuals, they call them cisses. They're cis, we're trans. I think it was just kind of a cute nomenclature, I thought. Oh, that was only me again too. Okay, I see. Cis and trans. Trans is opposite, cis is same. And anyway, but that also, I'm going to stop. There are segments on the DNA, they're called cis and trans segments, and they are biparallel or anti-parallel recognition sequences from strand to strand. And they're just there, and it probably goes to regulating replication or transcription. It actually probably regulates the DNA itself. So those are cis and trans segments of DNA that just probably control DNA. There are lots of repeat sequences, and that probably has all kinds of different functions to just identifying places where you might start transcription and stuff like that. So a lot of that. A lot of viral elements and transposons. Do you remember the word transposon, which is just kind of a little, not really a virus because they don't exist out separately from cells, but they are transposable or genetic elements that can go from DNA to DNA. And we really see a lot of transposon kind of elements at work during bacterial conjugation, a bacterial sort of ways. Bacteria are asexual cells, prokaryotes. They have DNA and it encodes their proteins. But everybody wants to mix it up a little bit with their DNA, and that creates survivability, which is why we do it in this elegant way, bisexual, by two different species that interact for mating. But if you're a single-celled organism, you still can get some DNA from the outside world. They can take it up directly from the outside world, or they can interact and trade off little segments of DNA. Those are transposons. Well, they exist in bacterial cells and in unicellular organisms. They exist in us as well. And so viral elements can be part of our DNA. Viral DNA viruses can incorporate directly into DNA. Some RNA viruses can, the retroviruses especially, that can become DNA. They can just integrate to DNA. And so 8% of our genome is viral elements and transposons. That is so wild. Don't we think of it as just like really clean and it made us and we all work and all that? There's a lot, there's this wild DNA seeks to protect itself, certainly, but it seeks diversity as well. And so there's a lot going on out there. So that is part of what our cell has to do, is to keep those proteins from being transcribed and translated as well. So that's part of the control and downregulation that maybe some of the microRNAs are doing. And then telomeres, mere would mean, telo meaning end or finish, and mere meaning body. So these are repeats at the end of the body. And so these are repeats at the end of a chromosome. And they're in the neighborhood of like 20-ish or 30-ish repeats. And it might just be sort of a protected little end cap in a sense, like if that's repetitive DNA and something can happen there and go wrong. We really hate for something to happen and go wrong in the coding sequences. That's just the non-functional DNA, so all of that. And another kind of DNA, so we have the DNA that is the 23 chromosomes, and then there is mitochondrial DNA, and you hear more and more about this in sort of, on the forensic shows, or all these shows, you know. I mean, if it starts out with a hiker came across a gruesome discovery, I'm watching it. And so you'll hear about mitochondrial DNA quite a bit. There are 37 genes in the mitochondrion. The mitochondrion is a double layer, double membrane organelle. There are several in a cell, and it's the powerhouse of the cell where the great majority of oxidative phosphorylation occurs, or the production of ATP. So, you know, you take in the glucose, you do the glycolysis, you do the Krebs cycle, and then you prepare it turn it into acetic acid, which attaches to that acetyl-CoA. That's going on that part happens in the mitochondrion, where you can then use all of that proton motive force, or all of that energy from the electrons that got released when you were breaking apart glucose, and you use that to drive the reaction of putting that last phosphate on, and you need oxygen to take those used up electrons at the end, so the cell needs oxygen. It gets rid of carbon dioxide because it burns the glucose. Oh my God, why am I talking about this? It's genetics, but see, it's all related. So the mitochondrion is the place where the ATP gets made. It's really where the oxygen has to be to receive electrons in the end of it. So your cells need oxygen, and we're going to talk about lungs next time, because the cells need oxygen there for the mitochondrion. And that's all kind of connected, right? So, anyway, that's such a little world in there, that mitochondrion, that mitochondrial DNA is there. And, you know, there's just a lot of people spend a lot of time thinking about what came first, you know? I mean, mitochondrion kind of a sort of autotropic kind of thing that was making its own glucose, or using its own glucose, making ATP, and got absorbed into a cell. I don't know, it doesn't. I don't really care so much, except that every cell has mitochondria. So that's a big deal. And it comes from the mother. So maternal, mitochondrial DNA shows a maternal inheritance pattern. And this is why, because boys and girls make the new fertilized egg. So what we've done is, it's called a fertilized egg because you really have egg. So what we've done as a species along the way is decided that we're going to have gametes. We're going to have boy gametes, girl gametes, or opposite gametes, polar gametes. And we've decided to do it with one great big gamete and one little bitty gamete. They're not iso in size. They're not the same. There are isogametes out there in the biological world, but we didn't do that. So we have a big fat egg and a little bitty sperm. And therein lies all of the differences between us. So she's making a few big, fat, delicious eggs, and then he's making a whole bunch of sperm. But what it means that he's only contributing DNA to that picture. And it makes sense. The new cell has to start out with all the goodies. So she's going to donate the goodies. That's where that comes from. So all of the mitochondrion, all of the stuff, that stuff, internal membrane, all of the organelles come from a maternal inheritance because it's a big, fat, squishy egg. Well, I don't know that it's squishy, but it's rich and thick and chocolate, you know? so that's why mitochondria come from a maternal inheritance. Yes, that makes perfect sense, doesn't it? Isn't it precious, really? Now you know, or maybe you didn't. Now you know again. How is DNA organized? Okay, it's really long. I don't know how many nucleotides are there. I wish I did. I think I wrote it down. That seems like something I would have tried to tell myself. I didn't. Anyway, bunches. Huge amount of DNA. So it has to, and it all fits into a nucleus. And so we have to have a lot of organization and packing. It has to be well packed. So there's some liver cells, and you see their nucleus, and you see that's a cell in prophase, and so the chromatin material is all in there. Okay, so DNA gets wrapped up into a wrapped state, or a semi-packed state called chromatin. And when chromatin is condensed to its fullest, DNA is called a chromosome. So DNA is DNA. DNA plus its associated proteins can be called chromatin or chromosome. So you need a lot of proteins to help package the DNA. And so a chromosome is so highly condensed and so highly packed that it's, one, no longer active for the most part. Nothing's happening with it in terms of genes and coding and transcription and so forth, because you're moving it. Everything's boxed up for moving when you see a chromosome. And it's so highly condensed that you can see it under a light microscope like you used in the lab during undergraduate, which is just phenomenal, really, when you think about that, how tightly that's condensed. And so, the DNA is organized into chromosomes, but active DNA is really in the form of a chromatid. Or I should not say, I should say condensed DNA is chromatin. So I'm going to write that down because I said weird words just then. Just normal chalk. Okay, so, oh look, chalk. I haven't used that in so long. Chromatin is really the state of loosely packed active DNA. So it's DNA plus proteins. Oh, you don't know. Every room has markers, and they're squeaky, you know? That sounds so satisfying. And then, if you condense it enough, you call it a chromatid. DNA plus protein, and then a chromosome. Now, here's the thing. This is a chromosome, and this is a chromosome. So this is a chromosome and this is a chromosome. We call this segment of the chromosome a chromatid, and this is its sister chromatid because it's an identical copy of this one. And they're kind of paper clipped together. There's a little section with some protein in it with centromere, meaning central body. You know all these words. But I feel like this is one of those areas where things get a little weird when you look at meiosis, because this is a chromosome and this is a chromosome and the material is identical. This is just replicated. The copy is attached. So these are sisters. And so, when you see DNA organized as a chromatid or a chromosome, it's so highly condensed it's not really active anymore, and the cell is undergoing cell division, either meiosis if it's in the gamete or mitosis if it's otherwise. Yes?
Is the chromatid active or?
Oh, you know, probably there's never enough. There's probably not completely off. Now during mitosis and this chromosomes are actually moving, those are probably truly inactive, but there might be a little dab of something going on. But we can say, no, they're inactive for the most part. So that was wishy-washy here in the political season. Did you guys get a chance to vote yesterday? I know you're all in this thing, and it's difficult to look out into the outer world. But anyway, that's okay. It's a primary. Just, you'll never forget voting for president, though. I can look back and remember all the choices I made. And I will tell you, I voted for Mondale Ferraro. You might want to Google them. No one ever heard of them the next day after that election. So anyway, but you just remember it. So anyway, I'll just throw that out there. So you've heard these words. There's a histone, which is a protein that helps wrap DNA, and then you get enough of those wrapped together, and that's a nucleosome. And this is how you wrap. So there's a twist built into DNA, as you know. And so when you twist something that's twisted, you know how it continues to knot on itself? The cell takes advantage of that, adds some proteins, and it's highly organized but quite packed when you see it that way. So it becomes very condensed. Let's see what else would I say from that picture. I don't know. I just love pictures of DNA. So chromosomes now, we'll talk about the function of DNA, but let's get to some chromosome information now. They're actually visible during cell division. So light microscopy, you can see them. Here is liver cell, and there are various states of mitosis going on. And you remember those phases, prophase, interphase, metaphase. It's just so lovely that you can see it happening. And it's actually quite a short process. And when you have that snapshot of a layer of cells on the slide, you can see it. Those are the chromosomes. There are autosomes, which are the 22 pairs of chromosomes. And then there are the single pair of sex chromosomes that identify gender. So there's, two flavors of those, XX and XY. And they're so named because of the actual appearance of the chromosomes. So a karyotype is a way to have a pictorial diagram of chromosomes. And this is looked at certainly a lot from a zoological sort of standpoint, or species standpoint. But in amniocentesis, karyotyping can be done just to make sure there's all the chromosomes, there's not an extra chromosome or a deleted segment and stuff like that. And then you can certainly get a real sure gender. Oh, we can't be sure of that anymore. You can know what the sex chromosomes are anyway. So this karyotype shows an individual that's XY. Oh, yeah, and genes are not evenly dispersed among the chromosomes. Some chromosomes have 100 genes, some have 1,000 genes. So how the segments got divvied up and such is who knows, it's interesting. Well, and the other thing I was going to tell you, every species, why we can't interspecies breed, is because there's a different number of chromosomes. The DNA is pretty similar between us and chimps, but they have 24 chromosomes in primates, and so that don't work. You can write that down, that don't work. Thank you. Okay, so this is sort of skipping forward with a word here, but I wanted to talk about some genetic chromosomal diseases early and then go back to some technicality about DNA. By the way, there wasn't a particular chapter anywhere I can refer you to. There just wasn't kind of one place because this sort of bleeds over into the sort of OBGYN section a little bit. So we're keeping it here for genetics. So you just have to use whatever text you have as a reference or the internet, and you can believe everything on the internet, as you know, because it's in print. Human chromosome disorders. So we're looking at actual chromosomes. So if you don't, not a gene error, not an encoding error, not a protein mistake, but what if the whole chromosome has an issue? There are a few diseases, and I just kind of want to give you insight into a few of those. And it happens, why chromosome disorders happen largely is because of non-disjunction, which just means things fail to separate appropriately during meiosis, or the formation of the gametes, eggs and sperm. And so I believe I added some information on this slide after you have it there, so I can There are homologous pairs, so we have 23 pairs of chromosomes. So mama's chromosome 1 and daddy's chromosome 1 are homologous pairs, and they need to separate cleanly. When they don't, that's called non-disjunction. Not junction, but not disjunction or pulling apart, but non-disjunction. And so if during meiosis 1, when the homologous pairs separate, you can have something go wrong and they not separate appropriately. Or you can have a failure of then the sister chromatids when that replicated chromosome then separates during the second phase of meiosis 2, and we're going to cover this, so don't wig. If that fails to separate well, you can also have non-disjunctions. There can be more than one. There's room for error throughout the 2 phases of meiosis, non-disjunction. You just end up with more or less than you're supposed to. That's another way to think about it. And I like to think about things very easily because I want to rely on common sense everywhere I can because it's hard to remember all of this stuff. And I believe I said something to you, like that to you last time. You already know a lot. Use it. And I put M1 and M2 there for meiosis. So here are, okay, look at me, not at the slide. This is just something from, I actually think this came from that Banov text. And it's just sort of a list for you to have there if you want to refer to it, going past it. So Down syndrome is our most popular in terms of ones we know about. Down syndrome, or a chromosome disorder, trisomy 21, meaning trisomy 3 bodies. So chromosome number 21 instead of 2, there are 3. And so it is Down syndrome, not Downs syndrome. And so I just have a little bit of information about it. I don't want to do a ton of information about these genetic disorders. We're staying in genetics and what is the problem? Where is the error? And that's what I want you to understand. And then add in the diseases as they come along. But, you know, Down syndrome has very characteristic sort of physical attributes, facial features, short stature, macroglossia, or large tongue. A lot of heart defects associated with it, susceptible to respiratory disease, shorter lifespan generally in the sort of special needs population, the lifespan is shorter. Sometimes the immune system is a little compromised in that population, prone to developing dementia, leukemia, often sexually underdeveloped, sometimes sterile. And that I put usually some degree of mental retardation. And I will continue to use this phrase, mental retardation, but because the R word has appropriately such negative connotations. The federal government has removed, MR used to be all the forms had MR dash and then a number for what form it was. So if it was under mental retardation, they removed that from all the forms. And they're using intellectual deficiency, so ID now. Because really trying to wipe away this retardation as a word, which was formerly a diagnosis and has been used, you know, as an ugly kind of slur sense. So, but it's still used for, in the state of Oklahoma anyway, on paperwork, and to describe it as a diagnosis. So you'll see it both ways, or in a lot of ways. A few other, and I've just kept this real short because these aren't patients that are going to be walking into your office, but Patau syndrome is trisomy 13, and it's one of these, a lot of the chromosome disorders are going to be fatal in that there will be spontaneous abortion or miscarriage. So what are the ones that get born? These are the ones we're talking about then, and so these do trisomy 13, serious eye, brain, and circulatory defects, often cleft palate. Of course, cleft palate isn't, you know, just with these kind of chromosomal disorders. The face and the body form from 2 halves and merge together. And so that last little bit sometimes fails to merge, and it has nothing to do with any sort of intellectual deficiency. Also, it's not called a hair lip. Please use the terminology cleft lip or palate. And in 1 in 5,000 live births is not uncommon. These children probably live a few months. The other non-fatal one is trisomy 18, almost every organ system affected, quite rare, 1 in 10,000 live births, and again, don't live very long. So our one, when I say most popular, it's not inconsistent with life, trisomy 13. Trisomy 21 or Down syndrome. A couple of other chromosome disorders, and I've always thought these were so interesting. Klinefelter syndrome is one where it's 47 XXY, so somebody has the, on the X chromosomes has an additional, the sex chromosomes, there's an additional X chromosome. And so these are males, but you have this additional X chromosome, so there's a little bit of kind of, I guess, confusion in some gender sort of specific things. And I don't mean confusion to sound, I just don't, I want to reword that because it sounds like a judgment or something like that, but there is more of a, maybe less definitive gender, is maybe what I want to say. Sex organs specifically affected, small testes, generally sterile, breast enlargement, other feminine body characteristics, often quite tall, men with kind of larger hips, and so there isn't any sort of intellectual deficiency associated with it. This is just some form of infertility, really, and maybe the only time it comes to light is during noted that someone is infertile. And so not inconsistent with a long life. Monosomy X, you've heard it called Turner syndrome if you've studied these before. Well, I didn't put the number for Klinefelter syndrome. Well, I don't know that I have that here. I don't think I do. I don't know the rate of Klinefelter syndrome. How many births is what I'm trying to say. I don't know. But monosomy X or Turner's, the only viable monosomy. So if you miss a whole chromosome, mono, 1, somy, body, 1 chromosome for that pair rather than 2, it's the only viable one. Because with X chromosomes, XX, you don't, what happens is you're really having 2 for every chromosome. And so you don't just turn a chromosome off. Remember, all your cells have 23 pairs. You don't just turn a chromosome off. So that really comes down to which gene is on. And that's the dominance and recessive thing. So for females, the X chromosomes are there, and females are actually a mosaic of expression of both of those chromosomes. So when you have only one of them, all the things are there. It's not not viable to life, but it does create this some especially gender-related kind of thing, which is interesting because why would you have 2? It really takes both, I guess, which is what's interesting there because they should be otherwise pretty identical. So with women with Turner's only have 45, not 46 chromosomes. So we say X is there and there's a 0 for the one that's not there, so XO. Genetically female, may not mature sexually, sterile, so it's kind of in sort of the female version of kind of what Klinefelter's in. Often have short stature, normal intelligence. But because a monosomy is so fatal, a lot of them fetuses don't make it to birth. So while there's quite a few, it's also monosomy is not quite a thing overall. And then a last one, or last couple, Cree-Duché, cry of the cat. This is because the infants have a very specific cry. I never encountered a birth with this. You may. There's just a small deletion in a portion of chromosome 5, not very consistent with life. These don't necessarily live a long time, but can live a few years. Severe intellectual deficiency, small head, just developmental issues. Fragile X, the most common form of mental retardation, really, that spectrum, the spectrum that we talk about, from autism and all of this stuff, so that encompasses everything. And it certainly encompasses autism, so a lot of Fragile X and autism. And so there's this repeat, or kind of these telomeres at the end of X chromosome. 29 is normal. There are over 700, and for some reason this creates this fragility of this chromosome. Part of it can fall off or break apart. And so, for whatever reason, you see this in, I don't think I wrote that right. I think it's more common in males, and I feel like I may have turned. So that number, I'm not going to ask you incidences anyway on the test. I should tell you, I've been asked to submit 7 questions for this, so you'll see however many on that test, 7 of them will come from this section, so that you know. I'm not feeling 100% about where it says that, about the incidence in males and females. So don't believe that. So anyway, I just wanted to show you a personal picture. That's my brother. He's never been diagnosed with Fragile X, but he certainly is profoundly disabled with mental retardation on his guardian. And he's older than me, and that's him. And that's an unusual picture of him looking. Now that we can do selfies, he's autistic. And that was kind of a later diagnosis because we weren't using that terminology in the 60s, in the late 50s when he was born necessarily. But it's hard to get him to look at you. He just doesn't do that. I mean, he knows me, recognizes, he follows commands, he'll come up to me, whatever. But now that you have this picture, that's kind of fascinating to him. So for the first time, I've actually been able to get these pictures of him looking at the camera. So, and he's amused by it, so that's that was at Christmas. So anyway, just wanting to tell you, this is a subject that's near and dear to my heart, and this is why I am so anti-R word. And you're out there, there are a lot of words that make upset people, and I know that. Sometimes, you know, you shouldn't be overly sensitive, and I've heard it all my life. But you know what? It's a big deal. This is how we communicate. This is what we do, we communicate, and words matter. And so, there you go. I'm not going to tell you not to say it, but I will say, if you're in the medical, I've heard a lot of medical professionals say, use the joke about Special Olympics, or that's retarded, or I've heard it a lot, and you just kind of go, wow, you know, you're a leader, and so, I don't know. Anyway, I think you're leaders, and you're communicators and educators, and so anyway, I would just have you consider strongly being careful about the use of the R word. So anyway, that's my brevy, that's Carrie. Nucleic acids. I'm going to just start this, give you a little break, and then come back. And you're going to like me a lot better today. It's going to go 4 hours. And you're laughing because you're not going to like me either way, right? That's all right. I'm not here for my ego, boo. DNA, deoxyribonucleic acid. The sugar is deoxyribose. Ribose is a 5-carbon sugar, and you see it in the little, what is that, penta something? Well, 5, penta. Yeah, it is. I knew exactly what it was. It says S for sugar, but you see over here in the blue circle where you can see the, what is that called? Skeletal representation? I don't remember. But there are 5 carbons in ribose, and you see 1, 2, 3, 4, and 5 is coming up here shooting off the little cyclic part of the molecule. And so that's that 5 prime, that 3 prime to 5 prime that everyone made such a big deal about. I never cared about that. And as much as I love DNA, I don't care. You go one direction one way and another direction the other way. Not a big deal about it. But that's where that comes from, as you well know, I'm sure. There's a phosphate group attached to that 5 prime of the deoxyribose sugar. And it's called deoxyribose because it has a less oxygen than ribose. Cute. And then there's this nitrogenous base. Base, what does that even mean? It's just a nitrogen-containing cyclic molecule, nitrogenous base, and you see it there with its nitrogens. So nitrogen then becomes a very massive issue for your body because you get enough nitrogen, you're having to deal with it because there's DNA and you're intaking foods and you're having to deal with nitrogen. It's also part of amino acids. And you end up with ammonia in cells that's toxic. So everything good is, there's some toxic aspect to it, just like oxygen. So this is a lot of, this is, you know, nitrogen is there, nitrogenous bases. And you know the nitrogenous bases then The sugar phosphate portion of the molecule binds to one another to form a sugar phosphate backbone. So you kind of see this yellow picture of that. And then the business end of the molecule, the letters on the page. The sugar phosphate backbone is the page. The letters are what you read, the business part of the page, are the nitrogenous bases. They array, they project away from the sugar phosphate backbone so that they're available, and they come in 4 flavors: adenine, thymine, cytosine, and guanine. ATCG. You know all that stuff, I'm sure. Bases, complementary base pair to one another. A's like T's and C's like G's. And so the 2 DNA strands then, they're anti-parallel because one runs 5', they're like this, they're anti-parallel. What's a better way to say that? Like a zipper. And so the bases complementary pair to each other. Oh, bless you. And it's cedar fever. Cedars are in bloom. And so the 2 strands are adherent to each other through this very it's an attraction, this bond to each other. It's not a permanent bond that's covalent, never the twain shall separate, but it's an attraction. So these strands can breathe apart, like Velcro. It's not a permanently closed thing, it can happen. So that's the beauty of DNA. So take a break until 11 o'clock, and then we've got some stuff to do in the next hour. Come on back, it'll be fun.